Thank you very much. I'll walk around, but I've got this for the video. I'm going to start off with a warning. You should never start a lecture with a warning. But there's some note. I had a note when I was teaching some second-year atomic physics a few years ago, and it opened up from this student that says, "I hate this lecturer." And in one sense, I hope at the end of that, for the reason that I'm about to give, you hate me as well. Because it went on to say, "I hate this lecturer because he makes me think." Now, actually, I thought that was part of my job, but obviously they didn't. And actually, if nothing else, if I've made you think about things in a different way, I've done my job. So, putting light into the life sciences. And I use the word life sciences so it covers medicine, biology, all aspects in there. Or as you'll see, looking for Nemo's heart. Now, actually, Nemo is a clownfish, and what I'm going to talk about is zebrafish. And I did once have a person in the audience that complained about that, so it's come up with its proviso. So, can we find Nemo's heart? So what I'm going to try and do is give you a little bit of a philosophy to begin with, and some historical background. And then we'll look at zebrafish and why they're actually useful for humans. And then can we watch individual cells and then go all the way through, and at the end of it, to show you how astronomy can play a very big role, potentially and in reality, in looking at biology. At least that's what I aim to do. So let's go back slightly and say, how does a physicist look at problems? And in essence, the first thing you do in any good bit of science is you image, you look at something, you observe it, you see what's going on. And then as a result of that, you try and build a model. So you go, "Oh, I think it's working this way." You can call that a model if you like. And then you sort of say, well, if my model's correct, if I push it in this direction, or I do something, I predict this is what's going to happen. So we do that, and then we do an experiment. So we perturb it, test it, we image it again, and you then keep going back and say, have I got this right? Is my model correct? And you refine it, and in a sense, that's the way that a physicist and generally scientists look at a problem. And I've put in the middle of this the 4D cell. And the reason I've put that there is, crucially, if you look at biology, it's a four-dimensional process. It's X, Y, Z, and time. And time is an important aspect in this. In fact, if you actually look at it and you try and apply problems as the way a physicist will look at a problem in biology, you've got quite a lot of scales here. So if we start off with a cell, things happen on the microsecond, about a millionth of a second. And the chemistry may actually happen even faster than that. But it's about a micron in size, and it's about a millionth of a second. And then those cells go up and build tissue, which might take hours, or it might be millimeters, and then we can go around and you can go all the way up to a whole organism. So if we take it in one sense, and we take what is going from a single cell, few microns, all the way up to a tree, and then if we look at the time that that takes, which is we go from millionths of a second all the way up to 1,000 years, or even longer for the way biology develops, you actually end up with the ratio of the scale of the problem you've got in 4 dimensions, time and the X, Y, and Z, is actually 1 with 27 noughts after it. That's quite a big range of scales that we're going to have to begin to think about. It dwarfs, in a sense, anything that people do in terms of astronomy and things like that. That's an enormous number. So somewhere, we're not going to have one model that solves everything, one method. It's piecing together a whole lot of bits of information. So it's worthwhile remembering. It's quite a long, and I think there are 27 noughts. I did actually take care on that. So how do we then approach this? I said there's a bit of philosophy at the beginning. The crucial bit, and it's true with any science, is you get the original question right. So someone comes to me and say, "Can you build a cheap microscope?" Yeah, I can build a cheap microscope. But that's not what you want. What is it you really want to watch, observe? I can build an instrument that enables you to do all that, and then we might go and speak to a mathematician who can then perhaps interpret the data, but they might say, "Oh, I can't do quite that. But if you could measure this," and the biologist goes, "Oh, I could do that if a chemist could alter this." So you put together the whole thing to try and understand the whole problem. And the crucial bit there is, and it's a cliche, there are no such thing as stupid questions. In other words, if you don't understand it, if you start working with somebody outside your field, it's not a stupid question to ask them to explain it. And the good people really can explain it very well. In essence, an awful lot of what I do, as you'll see as it comes through, is I either invent new technology, or actually I often go and steal it from somewhere else, because different thing, but I use the laser diode that's in a CD player, and we use it with a webcam to image teeth and to take X-ray-like images of teeth. It's because I understand how the physics works, and we can do this. And you can then choose something. The CD player is $1, pound, whatever the unit of currency is. So I can steal technology. The other thing is, the crucial part is you choose the right tool for the right job. So I could actually cut my toenails with a hedge trimmer, but it's not the right job it's not the right tool for fairly obvious reasons. So this all comes back to the same thing. Ask the right question and then choose the right method to try and solve it. And I hope that's what you'll see as it comes through here. I mean, crucially within this, people have often done this. So doctors have always used their eyes. So if you think about it, that's observing. It's using light. And there's lots of physicists who have played with it. So this is my eye, and that's Archimedes. I'm not sure whether he's a physicist, but he's a polymath. Only we don't always get it right, because what Archimedes thought happened was that the eye gives things out. It bounces off the thing it's looking at and comes back, and that's how you see. So clearly I should add a warning here. We physicists don't always get things right. Newton also worked on the eye and in biology, and this was when he went back to Norfolk during the Black Death. This is not an experiment to try at home. He got a leather bodkin, which is a thing you use for sewing leather, and poked it round the back of his eye and wobbled the back of his eyeball and looked at the way it distorted his vision. Don't do it at home. He actually got it right. It does distort it in that way. But the other thing is, any physicists in the audience doing A-level physics will probably have heard of Helmholtz, pair of parallel coils. Well, he did lots of other things. He actually invented this instrument for looking at the back of the eye, which is an ophthalmoscope. At the same time, another Luckensian professor, so Newton was the Luckensian professor at Cambridge. Does anyone know who that is? You might get it from this if you don't recognise the picture. It's Charles Babbage. So this is his difference engine. And Charles Babbage, at the same time as Helmholtz, also invented an ophthalmoscope. Now it's interesting, this one uses a mirror which is partially reflecting, so you can reflect a light off and look through it. This one uses a little hole in the middle, and those are still the two styles of ophthalmoscope that are used. Helmholtz actually patented his, Babbage gave his away. So that's given a little bit. So physicists have often played, in that case it's with ophthalmology, but they've done a number of different things. So let's think about microscopes. So these are early microscopes. We knew how to make lenses. Romans had some sort of lenses, but to put it in a bit of context, sort of about 1600s, Leeuwenhoek invented a double microscope. This is a variation. This actually just has a single glass bead on there, and he could get 200 times magnification. And he took some images. The sample actually goes on a little point here, and you hold it up to the light, and you move it backwards and forwards. He imaged things such as sperm and water daphnia. Now, how you get those to go on a point, I have no idea. So experimental detail is lacking. But there's a little bit that's depressing in the next slide. These are images, they're taken from Leica's website. This one up here is actually taken with a modern version of Leeuwenhoek's microscope. It's just a glass bead with a camera on the end, but rest's the same. That's a very modern, expensive Leica microscope. I'll choose a guess, it's probably about £25,000, £30,000 worth. This is a so-called dark field where what's white and black, it just runs it the other way around, and that's an electron microscope. Actually, they're still pretty good. If anything, the only thing that's really happened, and there have been lots of advances in microscopy, but the thing that's actually interesting is the core thing has gone on and it's been advanced. Probably the biggest thing that I'm going to use is the fact we can now record images electronically. You don't have to sit down and draw what you see. You can record it with a camera. But apart from that, actually the optics have undoubtedly improved, and you'll see these are obviously, we can't take three-dimensional images, as we'll see in a moment, and they're slow and all that sort of thing. But actually the pure simple bit of optics, we've got a bit better, but not phenomenally so. So what does a life scientist really want? They'd like subcellular. They want to see things happening inside a cell. So in round terms, better than a micron, better than a millionth of a meter. They'd like 3 dimensions. The other thing that we want to do is we want to make this minimally invasive. Because if I'm looking at something and I start chopping it into different things, it's sort of a bit invasive if it's biological. It tends to not do it a lot of good. So we want to make this minimally invasive. But I add a warning here. The moment I put light onto something, I am invading it. If I imaged my kidney has no defense mechanism for light because it never sees light normally. So you do have to really worry about how you're perturbing, how you're altering the system. You'd like to go and do it in depth, and that's where we come in. And we'd actually like to do it in vivo, because a lot of biology that's been done in the past, it's a bit like trying to follow a football match. So you go to a football match, and you dig a hole in the middle of the pitch, and you point your camera in one direction, and you take a picture at 1 minute. Then you go to a totally different football match, and you dig another hole in the pitch, and you point your camera in what you think's the same direction, and you take one at 2 minutes. And you do this 90 times, and you work out the rules of football. That's effectively what a lot of biology has to be done, because you do sacrificial bits of animals. Actually, how are you going to work out the rules? What I'm trying to do, and hopefully it'll come out, is I want to put one camera over a whole match and work out the rules that are going on. That's the analogy that you want to follow through. And this, well, not quite him. It's actually those things. Zebrafish are a really important tool in doing this. Now, some of you may keep tropical fish at home. It's the same zebrafish that you have in a tropical fish tank at home. They're basically transparent. They have some black pigment lines, but they're actually very easy to get rid of. You can actually breed them out, and that fish is called Casper. So it's a ghostly fish. It has no pigment. They develop very rapidly. They're easy to genetically manipulate. The thing I find most amazing is a zebrafish heart is basically a plastic pipe that sort of squeezes. You'll see one beating shortly. And your heart has 4 chambers. But if I take 24 drugs that work on your heart, 22 of them work in the same way on a zebrafish. Only 12 work in the same way on a mouse, which has 4 chambers. The other thing is zebrafish, it can actually repair its own heart. If it gets damaged, as you'll see, it can repair its own heart. I'm afraid to tell you, you all lost that ability about 30 weeks post-fertilization. A mouse loses it about 1 day post-birth. Somehow, zebrafish can repair it. It moves cells around, it changes their type. We can't do that. They're actually a very good model for looking at cataract development. They're a wholly good model for a whole lot of things, and I put the kidney in there because you'll see I come back to that. But we want to see what's going on. And an important technique here is a thing called fluorescence. So what happens in fluorescence is, I've got a dye in here. There's a different version I can have at some point. I've got a little fluorescent fish, but it needs a bigger display. So this is just a dye, a fluorescent dye. And if I shine, this will work even here. I need to get the right bit, that bit. This is a red laser pointer. There we go. If I shine the red laser pointer in there, you won't see anything at all. But if I put a different wavelength of light, so this is my green laser pointer, I put green light in, and what you can all see is some very bright orange, yellow fluorescence coming out. And this is now something that it means if I could use this molecule and put it in special places, chemically attach it to specific places in the cells, for example, I want to look at. I could even put 2 or 3 fluorophores in there and get them to go in different colors. And then I could get one that would be excited by the red, and this one, the red, will have no effect here. And then I can have another one that I put green light on, and I can then see it in the orange. So you can produce a whole spectrum. And this is a very important point, and you'll see where it's going to come into it. So in essence, for those of you with a sort of physics, chemistry background, there's my molecule. I put some green light onto it. It gets absorbed. So it goes to a higher energy state, sort of sits around there for a bit of time. But molecules, everything likes to get in the lowest possible energy state. You all like lying in bed rather than running around sometimes. So it loses a bit of energy, and then when it comes back down, it's lost a bit of energy because it's heated its surrounding up slightly, and then the rest of the light comes out. My green light comes out as orange light. It's lost a bit of energy, and it comes out at a lower level. So we can put one wavelength in, and the wavelength that comes out is a different color. And we can actually do this with different materials. So we could put chemicals into the thing. But there's a material that was really discovered, and it revolutionized the way biology happens. And you'll see how we're going to use all these techniques in a moment. It was developed by someone called Roger Chen, who unfortunately died this summer. He was 60. He won a Nobel Prize for this. He actually won it for chemistry. So he took the gene from a jellyfish. So this is the thing that produces, it's in the jellyfish's DNA, and this gene, this sequence of DNA, tells it to produce a particular chemical, and this chemical is fluorescent. This is the jellyfish, it's called Aquiori victoria, and it fluoresces. If you put blue light on it, it glows green. And he then took that bit of DNA and would deliberately put it into other organisms. So this, for example, is a zebrafish, and this is now tagged on to some particular protein. So when the jellyfish produces a particular protein for doing something, might be for producing a cell membrane, it might be just produced in heart cells, it produces this green protein as well. So we can now deliberately target, we've now got something that's inside the jellyfish, inside the fish. We can do it in mice, but again, the zebrafish is nice because it's transparent. And now, using my imaging techniques, I can now see exactly where a chemical is being produced and what's happening. In fact, you can take this to ridiculous extremes, and you can actually take the fluorophore from coral, and you can manipulate these things, and this was done by someone in Roger's lab, and they produced this fluorescent protein art. It comes from California, so perhaps that's why it's that sort of scene. So I've now got something that I can put in my animals and make different bits of the cells or different cells begin to light up. Right, quick recap. I've got a good model. Zebrafish is a really good biological model. I've got a biological marker, this so-called green fluorescent protein. I should add that the others used to be called red green fluorescent protein, and you had blue green fluorescent protein. They have at least now called them different names. If you look them up, you can have cherry, you can have raspberry, you can have strawberry, and I can't remember, the other side, blues, they called cyan yellow. They're a bit boring, but tomato. And we want 3D images. We want to do it in a live animal with minimal disturbance, and we want to do it for an extended period of time because we'd like to see things growing and develop and try and understand what's going on. So let's change track. Let's have a bit of optics. So the technique that we want to use is I want to produce a three-dimensional image. And biology, some of it happens very rapidly. As you'll see in a moment, a zebrafish heart beats about 150, 160 times a minute. And I'd like to see that. So I could sort of scan a laser beam and take an image, but that's quite slow. But I want to produce three-dimensional images. So the way that we do this is this technique, Single Plane Illumination Microscopy SPIM. We're good at making up acronyms. And what we do is we take a laser, could be any light, but we tend to use a laser, and we turn it into a long, thin sheet. So we produce a sheet of light, which we bring in from this direction. So we only illuminate a thin slice of my sample, and by thin, it's about a micron thick, thousandths of a millimeter thick. And we look at it in this direction. So we've taken an optical slice, and then we can either move the sample or move the laser beam and do the next one, and the next one, and the next one, and the next one. So I can very quickly take a whole slice in one go. And as a result of that, we've now got three-dimensional slices. I can begin to put them together. I can do it very quickly because I've got a camera down here, and we can run cameras up to 1,000 frames a second fairly easily. We're actually only putting light into that plane, so there's always a risk that we could damage the cell, phototoxicity, the light could kill the cell. But I'm only doing it in that lane, and these up here aren't being illuminated, so we've minimized that. The other thing that can happen is if I put lots of light on it, this fluorescence disappears. It's called bleaching. Actually, we've solved that problem because it's only this layer, and I'm only doing it with a quick flash of light anyway. And it actually, because of some various things, as you'll see later, the image, there's another technique called confocal microscopy where you scan a beam across it, but that degrades as you go more deeply. This does. The other thing is this is really cheap. If I buy a confocal microscope, I think a Zeiss one is about £250,000. These things here, the two microscope objectives are about £3,000. You can actually do the rest with a webcam. I've built one of these for demonstrations, just using webcams and some other optics. So if you're going to do it seriously as a scientific instrument, you can do it for about £30,000, which for a microscope is cheap. And so this is what we're doing. We're going to take a slice of light going in this direction, and we'll image it at 90 degrees. The other thing that we've actually got, it's not shown here, and you'll see why I need it later on. We actually have an LED that produces infrared light. So that's light at too long a wavelength. You can't see it. The camera can see it very clearly. And we can feed that light through to actually also get a transmission image through the whole animal. You'll see why I want that. And this is what it looks like. So here's the infrared light going in this direction. Here is my thin sheet of blue light. And the zebrafish is mounted vertically. We anesthetize the zebrafish with a very, very small quantity of anesthetic, and it's then held vertically, and they develop perfectly normally. We can keep them, other people have kept them for 4 days anesthetized, and they develop normally. So we're not perturbing it too much. And we image in this line, and the image goes off here. So the fish isn't swimming around. I'm not chasing this thing around. But these fish I'm talking about here are under 5 days old. They're embryos. So they're slightly active by the time they get to 5 days. And so we can begin to take some images. So this is the zebrafish brain. And these are the blood vessels in a zebrafish brain. That bar is, I can't remember what it is on this one. I think it's 50 microns. No, it's 50 microns. So we can take a whole slice of the brain in one go. So these are blood vessels within the brain. Or we can actually go somewhere else, and we can look at the blood vessels in the tail of the zebrafish, which if I click here, should start to rotate. So we can build up these three-dimensional images. So this was taken slice by slice and then reconstructed. We can actually do the same thing in plants. So this is now the internal, what's called these are the cell walls and the cytoskeleton of a plant. And I've lost the mouse for a second, but if we go back down here. So this is actually a plant root, and you can see how the root has developed, and we can then take these images and actually then reconstruct them. These are just pretty pictures that we put together, but these are actually valuable. And just as a comparison, this is a so-called different way of doing it. It's a high-speed confocal microscope. These are the images we get. Those are the images they get. So taking this one slice at a time means we can do things very rapidly. I won't play this one. There's a picture there of some things moving around. If people want to see it afterwards, they're actually inside a plant, but the contrast isn't that great. But we can actually then do it in an animal. So this is a zebrafish, and this zebrafish is genetically modified to produce cancer. And these green cells, they're producing this green fluorescent protein when they are cancerous. These red cells that you'll see are so-called neutrophils. They're the things that are in your body going up trying to kill the things it doesn't want. So this is taken from inside a live zebrafish. It drifts down slightly because it moved slightly, but you'll get an idea of these things moving around. There's one here going in and out of cells. There's this thing moving up there. So this is the neutrophil. This is the thing that's trying to kill the cancerous cells. And you can actually see it going through and beginning to go inside. So these have been labeled with a red fluorescent protein and the other ones labeled differently. So we can actually see the same thing. It's in a little bit more here. So this is, if you like, cancer actually taking place and your body trying to correct it. Whoops. Should go, come down. Is it? So again, now you can actually see here, this cell is actually sending things out to try and see what's going on. It's trying to sense its environment. I'm not going to go into the details there. It's not the area that I particularly know about. But we can actually then zoom in and look at some of these places. This sounds as though I'm boasting, and I don't mean it to come out that way. You are probably only about probably thousands of people that have seen these images. These were actually taken about 3 months ago, and they're in the process of going into a paper because the people are understanding what's going on. So actually being able to see the neutrophils, the thing in your body, okay, it's the zebrafish, actually going in and seeing what's happening inside the cells, that gives you an idea of what's going on here. I've lost my mouse for the moment. Let's go up one. That's it, reappeared. So if I come up to this one here, that's it. You can actually see now, if you look up, hopefully, this is a high-risk opportunity here. I'm going to go into low intensity light here. The audience can go to sleep now. So you'll actually, as it looped around again, no, this one's moving. I'll just go back and play that one again. Let's come back. You can see in detail how this thing is actually moving and going into that particular cell. And then you can actually see the same thing again on this one, moving around. And you can see, so we can actually study. So crucially, what I want you to get over here is, these are things happening. This is marginally speeded up. But you can now see what's happening in real time. So if we wanted to now say, well, we've got an anti-cancer drug, we can feed that into that system and see how it actually works. And we're actually doing this beyond. But can we go beyond plants? So I mentioned the heart. So I'd like to see an imaging, beating zebrafish heart. So how are we going to do that? So the way you would do it in a human is you'd attach an electrode to it and do an ECG. The zebrafish heart is about, at this stage, 200 cells. It's not easy to do an ECG on something that's only 200, and that's going to perturb it. I'm putting a needle in, and that's most of the zebrafish's heart. So what we do is we take a whole series of infrared images. So remember, we're just looking through the heart, and we analyze those images. We actually use an algorithm called SAD, which is the sum of absolute differences. We just compare each image. And we analyze those images, and we can begin to get to a point where we can predict when the heart is going to be in a certain position in its cycle. So this is this SAD index up here, and it goes up and down. These are the values of this SAD index, and you can see that this is the beating zebrafish heart. As I said, it's going about 150 beats per minute. And we can put these crosses in, and we can join them all up, and we can then use an algorithm to analyze down here. Now, what we actually want to do is take an image when the zebrafish heart is in a particular point in its cycle. And then we actually want to take lots of images at different points in the cycle and turn it into a 4D moving video. So we can predict when it's going to be in the right place. So we image it with the infrared light, perfectly safe. Just before the heart's going to be in the right place, we open our camera shutter, we put the sheet of laser light in, turn the shutter off. We've taken a snapshot image. And we can go on, and this is actually what happens. So the image on the left, this is the zebrafish heart in the infrared, and this is continuously. What you'll see this move, and you'll see the blood going through it, the blood cells. And what you'll see over here is the synchronized frame. It's actually dead boring, because all you see is the heart just seems to sit there like this. They're actually taken at the same time. You see the blood cells change position, but you'll see that we've got the timing such that, should start going, whoops, come here. So there's the heart beating. This is slowed down. And you can see the blood moving around, and you can see the wall. This is the one here. So you look at the wall, and it doesn't move. The blood cells you can see moving. So we've now synchronized it, so effectively we've frozen the way of doing it. The way people did this previously in the zebrafish is they put a drug in that stopped the heart, took the images, and then started it again. I think I would define that as perturbing the system slightly. We can do this. And we can track it for a long period of time. So we have actually imaged these fish for 24 hours as the heart develops. There is an interesting question and an aside. We also do this in the eye. That produces 2 terabytes of data. It's an interesting question of A, how you save it, and B, if you then want to copy it to something else. So data becomes a big problem with this, and that's just from one sample. And typically here you want to do about 40, 50 repeats. It's quite a big number. Get into hard disk production is probably the answer there. So how do we do that? We can take this image and we can synchronize it, and we can take our science image. So we're taking this rapidly moving thing, predict when the heart's in the right place, and then take a science image. Or we can actually use an ultraviolet, so a laser that's got a very short wavelength, as you'll see in a moment, which will damage individual cells. So we could potentially take them out. And there are also some techniques for doing very, very, very high resolution imaging. So this was the type of technique that won the Nobel Prize for Chemistry in 2014, so-called super resolution. It's not what I'm going to talk about, but you can actually image things very, very, very much shorter than the wavelength of light by playing some clever tricks. I realize there's one other thing I didn't do here. So what then happens is we take these images, and we can take a 3-dimensional stack at different, initially just at one time point, and we produce an image. So in a moment you'll see this is a fluorescent image of a beating heart, only we've timed it so it isn't beating. This is what happens when you give a video to one of your postdoctoral researchers. And I gave him this video, and he said, here we go, it's the 3-dimensional image. And I said, yeah, it's much easier if you get it rocking. So he played with it, and this is what came back to me. I think it'll work here. When sorry, I've overshot. I need to come back. So this is what happens when you give somebody and say, turn it into a rocking video. I have no idea what that music is. I'm not impressed, but this is... Right, let's turn it off. If it had been a bit of bark, it would have been all right. So this is now a 3-dimensional. So we've now got, and in fact, this is actually quite an old image. We can actually individually identify the individual cells within the thing, or we can put them together. And this is now, let me find the video. Okay, a 3-dimensional imaging of a beating zebrafish heart. So we've sort of taken a slice through it, and you'll see it, that's how it beats, moves, that's the valve at the back there going backwards and forwards. And we can now take a 4-dimensional, 3-dimensional, but we've now got the beating. We can actually track where individual cells are going. That's good. So we can watch how they move, they track how drugs go in, use high throughput screening, for example, for looking at different drugs. The other thing, I made a comment earlier that a zebrafish can repair its own heart. And this is clearly something, can we find out how a zebrafish does it, and does this then mean that there may be a treatment for humans and heart disease? So this is actually zoomed in on the zebrafish heart, and you'll see in a moment there's a little red spot there, and that's the valve of the heart. And what happens is we fire a UV laser and we kill a few of the cells at the center, at that valve. And then we watch how the heart repairs itself. Now, the way they used to do this before is they'd be seeing this heart beat, and it beats about that speed. And they go there. I've taken all the fun out of it. And they go, yep, oh, right, we've hit that one. Move on, we'll try another one. So it was like a computer arcade game. Whereas here, we can now effectively have what looks like a stationary image because we just present the image, and that means they can then take out the cells much more accurately. So you'll see this. should start going. So you can see the blood flowing through it. There we go, and we fired it and damaged the valve. So it's altered the blood flow. It repeats again. Here we go. So it's going all right. There's blood going through. Blop, the little thing that you see is a few of the cells dying. So we actually now have a way that we can take out a few cells. And in fact, this is what was done. I'm not going to go into the details, but this is, you then watch how the thing recovers. And the crucial bit here is the white is the control. In other words, that's the animal that we didn't do anything to, but we make measurements on it. The black one is the actual fish, or it's a number of fish that we damaged. And you can clearly see this is over, so that's before, that's 2 hours, 24, 48 hours. And you can see that, I mean, in this case here, I can't remember what that is, blood flow. 2 hours, it's gone down, but it's recovered. So you can see here that this zebrafish heart, we've killed a number of cells. If I did this on a human baby, the equivalent, the baby would die. But somehow this zebrafish has a method for moving the cells around and repairing the damage. So that's the sort of thing that by using some optics skills, some physics, the bits that we've put together, the biologists can now actually begin to look and try and understand what's going on. But we actually have another way of doing this. And this is, again, what you're about to see. I don't know whether it actually got submitted today. I haven't checked. We've sent it to the journal Nature. So there's also a way, I said we can genetically modify things to put a fluorophore in there. Well, actually, we can genetically modify them to put another compound in there that when we hit it with light, it fluoresces briefly, but then falls apart. And when it falls apart, it produces what's called reactive oxygen. It's oxygen, but it's really energetic oxygen molecules, or oxygen molecules, and they will kill things. In fact, the cell doesn't like it, and it starts killing itself off because it goes, I shouldn't be producing this, I better shut myself down. So you can actually, therefore, in certain cells, get them to produce this compound, and if we then hit them with light, those individual cells can be killed, and we can watch the process going on. And we do this, normally this had only ever been done in individual cells on a plate, and it's done with epifluorescence. In other words, we just shine a big red light on it. It's a green light actually on the top, and you know that some of the photons are going in and they're going to kill some of the cells indirectly. We used our SPIM and what's called a Bessel beam, which is, if you take a normal beam and focus it down, it produces a small spot. If you have a Bessel beam, it produces a long, elongated spot. It's like a dagger of light. It's a sword of light. And we then quantified how much fluorescence was coming off. So this is what we have in our system. So if you come back, if you remember, we have our light going in here, which is doing our imaging. So we've got a sheet of light in here, and we have our ablation laser going in and coming in through this. It's going off a thing called a spatial light modulator, which is just something that we can put a pattern on it, and it will produce exactly that pattern of light. It's an adjustable optic. And then we can image with different cameras in here. And so this is what it looks like. This is at 10 seconds. So this has got green fluorescent protein that the whole kidney is producing, and certain cells that produce a compound called renin produce this red dye as well, this so-called killer red it's called. We didn't give it the name. And when we hit that with greeny, bluey greeny light, that starts to disappear, and so the fluorescence has gone, and we therefore think it's killing the cells. In fact, you'll see that we activate it, and you'll see that happens. This is what it looks like when you do it on a lab bench. So here's my camera. We annotate it slightly. So this is the blue light sheet. The blue light, which is giving us our background image, comes in from that direction, and our killer beam comes up the table off this optic spatial light modulator into here, and we shine it in here, and then we look at it with the camera that's sat down there, and we then see a pretty picture. So our fish will be sat here. This is our imaging sheet of light, and this is where we're looking at it, but we're also sending in this greeny yellow beam to kill individual cells. And this gives you an idea. It doesn't matter. This is what happens if you just do it with a big, broad light. You kill it. You'll see in a moment that's not the only thing you kill. Here, we can now target just small areas. We're actually putting in a really low power. This is probably It's about the same amount of light I've got in this laser pointer. It might actually be less. But we can see here that the fluorescence has gone, so we can quantify how much is being produced. These are other ways. The other thing, we actually make sure that we really are killing those cells. So we do what's called a tunnel stain, which looks for the bits of DNA inside it. So when we've killed it, the way the cell kills itself is it chops up its DNA. And this stain looks for bits of chopped up DNA. So we can say, yep, those are the individual cells that we're targeting. And you can actually plot, so this is the intensity of the fluorescence over time, and it goes down in using different methods. And this is probably the important slide. If we do this with zebrafish with epifluorescence, basically they all die because it's just too phototoxic. The light kills the whole of the fish. They're really not happy. But if we use our beam that's coming in from a different direction, actually 100% of the fish survive. I should add that this defect that we're creating in the kidney actually causes a longer term defect in the blood vessels, which nobody has any idea real why at the moment. But that's what happens. And so then we're then looking to see how does the body repair it. So by using, again, these optics, we've now given the biologist a really good tool. But I said we wanted to go in 3 dimensions. And that means, well, 4 dimensions. We've got the time, we've got 3, but actually we'd like to look really deep inside the sample. And this causes a bit of a problem. This is not really a biological sample. It's Philadelphia cream cheese. Actually, it's not a cheese at all. They basically take fat, water and protein whey, mix it all up, and that produces your cheese, which are little balls of fat surrounded by protein which sticks the water to the fat, and so you get Philadelphia cream cheese. It's why if you leave it out, it goes watery on the top. It's called syneresis. These were actually done for Unilever some time ago. At the top, you can actually see it's got little balls of fat. This is just taken with it happens to be another technique called 2-photon microscopy, but it doesn't matter. It gives us the ability to image at depth. By the time we get to 25 microns, it's getting a bit blurry. By the time we go 90 microns, I cannot make anything out of that. And if you think about it, it's because I look at the top, that's all right. And now as I look at this layer, I've got to look through what I've just looked at, and it starts to blur my image. So the deeper I go, the more blurry it's got. We can actually show this. So this is, and it's due technically, what changes light, it's the refractive index. It's the speed at which light goes through something. So in air, it goes pretty well through undilated. You know, if you put a glass, if I take this glass of water in here and put the light in, it's going to get distorted because there's a different refractive index. Pencils look bent and things like that. And crucially with this, as you go through more and more changes in refractive index, it gets more and more blurry. So this is going to be an image. This is now a single slice of a zebrafish heart, and it moves around a bit. It's beating. And you'll see it goes a bit wobbly. Whoops, it comes back. Now we've gone to a different depth. And you'll see as we go deeper in, this is going to get more and more blurry because I'm now looking through more and more of the tissue. That's a problem. Got a question now. What's the favourite, I don't know how to phrase this, what's the astronomer's favourite nursery rhyme? Thank you. Why do stars twinkle? Those in the audience, certain members of the audience here are not allowed to answer that question. There's a slight cheat in what I'm about to say, which we won't go into. But why do you think the stars twinkle? Yeah? Absolutely. It's not the light flashing, it's the Earth's atmosphere. It's why you get very good images from the Hubble Space Telescope. You put it outside the atmosphere and the atmosphere doesn't get in the way. So this problem of looking at the stars, effectively it's exactly the same problem that I've got looking down a microscope. I'm looking through the layers, exactly as you said, of turbulence. The slight difference in the atmosphere is it's constantly moving around. In my zebrafish, it's not moving in quite the same way. So we have the same problem that astronomers have. And what they do on these big telescopes is they look at the star, and they have a special mirror that they call adaptive optics. It's a mirror we can change the shape of. So if the atmosphere bends the light in one direction, they can put the opposite shape on here, which counteracts that shape. Now, the way they then do it, they actually look at the shape of the light coming back with what's called a wavefront sensor, and that tells them what shape to put on the sample. So this then turns this galaxy. That's what it looks like without the adaptive optics. That's what it looks like with the adaptive optics. In fact, using a telescope with adaptive optics, you can now get better images than you can from the Hubble Space Telescope very often. And you may or may not be aware, one of the earlier lectures, you may have heard of the thing called the ELT, which is the European or Extremely Large Telescope, which is going to be a telescope. They've already blown the top off the mountain 6 months ago in Chile. They literally blew the top off a mountain in Chile, and that's where they're going to build this telescope. And the telescope is 39 meters across, so it's bigger. The mirror's basically bigger than this lecture theatre, roughly, about the same size. There's lots of little ones. The only way that's going to give you performance is by using adaptive optics, exactly as you said, to correct for the atmosphere. So the crucial bit is here, what shape do we put on that mirror? Now, the astronomers, very often, what they do is they launch a really high-power laser, either a green laser or sometimes an orange laser, which lights up some sodium, and they create an artificial guide star. Now, actually, the power that they put in this laser is between 50 and 100 watts. That is, trust me, that's a very powerful laser. If I shone that on my zebrafish, it would be very bright, but only for a short time. And imaging through smoke is really difficult. So we're going to have to be cleverer than these astronomers. There's one other thing I should say with this. When they build a telescope, the new extremely large telescope's about 1 billion units of currency, dollars, euros, pounds. It's about the same now. And I gave a talk on adaptive optics in America a couple of years ago. And the person before me was from NASA. And she had a mirror that she was doing the adaptive optics, which was an array of lots of different mirrors. And each one of them, I have to get the numbers right here, cost £10,000. And her array was 100 by 100, which if you do that, as long as I've got to remember the numbers right, her mirror cost $10 million. Sorry, I'm cheap. Ours are about £1,000. We can't use, it's playing a slightly different game. But how are we going to work out what the shape is? Well, actually, there might be a way of doing it. So what we can do, if our sample has lots of little bits of fluorescence, instead of putting a sheet through and lighting them all up at the same time, perhaps we can put a beam in from the side and just illuminate one, which we then come back. We put a pinhole, so in case we illuminate, some of these here might get illuminated, but that's the one we want. So we can put a pinhole in, so all we see is the light from this. And we can then split it up and put it onto what's called a wavefront sensor to tell how much that light is distorted. And in fact, we can do that in a zebrafish because if you'll see here, actually the zebrafish wall at certain points is made up of lots of little dots. They're the cells that are producing this green fluorescent protein. So they're like the stars. It's an artificial star. So we can actually do this and take multiple data points, this heart's beating, as it says, about 3 hertz, 150, 160, 180 times a minute, and go on. So this is now, you'll see this, where's the mouse? This is now the heart, and you'll see it beating near enough in real time, and you'll see that we can see those points as they move. That's about the speed that it beats at. And it's going backwards and forwards. And so we can actually image at high speed exactly where those points are, but we can also look to see how much, as we change the height at different points in the heart cycle, we can actually then plot what the aberration, how much the light's getting distorted. And in fact, you can see that this follows the heart cycle roughly. This one, which is focused, doesn't have much. Won't worry, this is how much the beam's been distorted in 2 different ways. But you can see that clearly here this is being distorted. So if I can now measure my distortion that way, I can actually then set something up. So here is my laser, which is producing now a sheet of light. The light comes off this way, goes off the digital mirror and goes into the camera, and this is the deformable mirror. This is the sort of cheap mirror that we've put in the corner. It's got little actuators behind it. We apply a voltage to it, and it pulls a very thin metallic membrane towards this mirror. That's why it's cheap, and we can change its shape. We actually put a laser on here so we can measure its shape. And we could use those little laser guide stars. We now actually know, because of seeing how much distortion it's got, we actually know what some of the aberrations are. And in fact, we didn't use those spots to remove the aberrations. We found it was better to use some algorithms. So we used two different things. So this is to choose what shape I put on my mirror. And the crucial bit is I need to get it right, because actually with this I can take a football and turn it into a rugby ball by putting the wrong shape on it. So I need to be really sure I get the right shape on my mirror. So one we can do is we can use a hill climbing algorithm. So we can look at my image, perhaps just maximize its brightness. And we can look at it, look at the image, and keep changing the shape of the mirror until it maximizes the brightness. And we can do this with a so-called hill climbing algorithm. So what that does is it marches along and it maximizes the metric. So that's all right, I've got to the top, I've maximized my brightness. Well, this is very good, but that is not the total improved performance in this hill. He's reached the local maximum, but he hasn't reached the ultimate maximum. So if you use a hill climbing, it will reach a maximum, or a minimum if you want to be a valley faller. But you can set it up to reach, but you can't guarantee you're going to reach the absolute maximum. You can also use what's called a genetic algorithm, and what that does is it takes the shape on the mirror. It takes about 3 or 4 different shapes, looks at the values and says, well, that mirror shape's quite good and that mirror shape's quite good, so I'll combine those two shapes, I'll breed them and get a new shape. And then with that new shape, I'll breed that with another good shape. So literally, you choose, it gets called the genes and the diversity, and you can actually then start to look at all shapes. And in fact, in that case, all the time, you get to the maximum. So this is now the other way of doing this. So what we would do, take our image and try and maximize the brightness in our image by putting different shapes on the mirror. And I've lost the mouse again. There we go. And now, so these are my mirror shapes, and I bred the best one and came up to here. Oh, it came over here now. So I choose that one and I choose the best genes. And you can now see that actually, if you keep doing this, eventually it reaches the absolute maximum. By choosing the best, you put several shapes on, choose the best 2 to breed together, you actually explore all the way. So this is the way, it's used in other methods. So using this, we can now actually optimize and produce a good image. And this is, when you look at this is actually a little bead. I think it's 3 microns in diameter. And that's what it looks like when you look at it through a zebrafish. When we put our correction on, we've nearly brought it back round to its small shape. It's still a bit distorted, but it's a lot better than that. And this is actually the metric. It doesn't matter. I think this one was done on brightness, but you can maximize. You can see as the different mirror shapes go on, so we get different things, and it comes out to be a maximum. We can actually model this as well. So this was actually what we think we would get for different depths looking at this thing, because we can model. I said earlier we could measure what the distortion was in the heart, so we can actually then begin to model it. So we have a method now of working out what shape. Crucially, we don't then need to do this constantly, because once we've worked out what the distortion is, it's not going to change rapidly. It's not like the astronomer's problem where you've got high-speed winds and things. And so we can put these bits together. And this is an image that you'll see going through Z. This is actually a fin of a zebrafish. We use it because it's nice and thin and really clean and easy to see. So this is the image with no adaptive optics. This is what it looks like when we put the adaptive optics correction on. And I will go through here. Oops, there we go. I'll just stop it at a random point. So you can see here, this is what it looks like without putting this correction on, and here it's very, very much clearer. We can actually then do something. So we've now removed this blur. So we can really now look at depth. So wouldn't be in the case of a zebrafish, but we've done this in other samples where we've used a microscope to image about 2 millimeters, 3 millimeters. into, happens to be a piece of kidney. So we can actually then begin to look at what's going on biologically, genuinely in 3 dimensions, and because we've got this ability to do it rapidly, we can actually then do this 4-dimensional thing and really begin to understand what's going on. Hopefully, I've missed it out here, I've made you think. Hopefully, that's gone through your head. I've hopefully also given you an idea of how to tackle really challenging problems. You have to bring together all the skills. So the work that you've seen here in the background is the skills of clearly the biologists to understand, to do the genetic manipulation. There are chemists involved in developing the fluorophores, some of the other bits. There's mathematicians involved in doing some of the algorithms, actually trying to analyze the blood flow. Different thing, haven't spoken about it, but you bring in all these skills, and then in my case, the optical toy maker, to put together these things, to really begin to understand what's going on, and how by stealing technology from, in this case here, some of it's from, some of it's from the video industry. The spatial light modulators are used in the video games industry, and there's one in that, slightly different versions in this digital projector up there. It's also, we've used the adaptive optics from astronomy on the cheap. I've also hopefully demonstrated why the humble zebrafish really is a huge advance, and how by doing these different things, we've talked about taking a slice of light to build up a 3-dimensional image, you can heart synchronize it. So we can then actually look at what's happening in real time. We're really trying to analyze what's going on. And we've done it minimally invasively. These fish survive the procedures that they're going through. We've also shown how you can optically target individual cells by genetically manipulating them, then hitting those with some light. You can optically take out individual. There are variations of this where you can actually then turn cells on and turn cells off. We were deliberately trying to kill them. So there's a technique called optogenetics where you can switch cells on and off. And we've also then showed that you can actually do, if you like, astronomy down a microscope. I have to end with the slide so at Durham, Laura is a postdoc that's still working on these. These are the people that do the work. You learn as you move up the food chain. I'm not sure whether you move up or down the food chain in a university or in any industry. You get less and less time to do the things that you actually quite enjoy doing. You have to do these in your spare time on a Saturday morning. Laura can spend the time in there. Johnny did the heart synchronization, is now a lecturer at Glasgow, and Chris is worked with Johnny and did some of the other imaging, has a lectureship in Durham, and Mariana was a Brazilian postdoc that came over on a fellowship, and she's now back in Recife. The other person I really have to say here is Charlotte Buckley, who was a physicist when I first went to Durham, and she worked in my lab one summer and said, I quite like doing this playing with biology. We were doing something completely different. She went and did a PhD in Edinburgh, actually, with John Mullins, and she's now developed all these techniques. So her PhD is in physiology, but her fundamental training is in biology, is in physics in Durham. John, and then Seb did the breeding of these fish. And then the funding I get, these people just pay me to have fun. Well, actually, they pay for these people to have fun and me to have the hassle. The British Heart Foundation pays for quite a purse of this for very obvious reasons. That's the engineering and physical sciences. We have money from the Leverkusen, and CNPq is the Brazilian funding agency. So I hope at the end of this, I've made you go away and think a little bit about some of the things that are going on, how you can bring all these different bits together. And at the end of it, I hope you've had an enjoyable time as well. Thank you very much.